So, so our talk, this talk will be different compared to many others. So what we've been trying to do now for several years is that we want to understand how the structure works. So we want to go beyond the chimera morphs. We also want to go beyond just keep determining structures, because as it stands today, Google will solve all the structures for us, or predict very soon. So we're looking at two states of the contractile tail assembly called pyocin, and it's produced by Pseudomonas to kill other Pseudomonas. So there's a very much like a typical phage. You can imagine a capsid here, but there's no capsid. So what these particles do, they go and kill other Pseudomonas. So what experimentally easy to capture for us are these two states: extended pre-attachment state and contracted post-attachment state. But in reality, there's a lot more states. And so what we'll discuss today is how we can and can we actually describe this many states. So here's we see particle is free in solution, and then it finds its host using this receptor binding proteins that can be replaced, and the particle can be retargeted to bind and kill other host cells beyond Pseudomonas, besides Pseudomonas. So there's something happens in the base plate that triggers this contraction process, and there's this point of no return, the transition state, where the particle has to continue to contract, and then it pierces the host envelope, opens the cytoplasm, the ions release, and the cell dies. It's one of the simplest contractile tail-like structures, and it's pretty small for this organelle. It's only 12 megadalton. Now, in terms of free energy, we know this is a high energy, a metastable state, and this is low energy metastable state. So these two, again, very easy to capture for us experimentally. And why is it a high energy metastable state? Because if you keep this particle long enough, all of them will be like that. Or you can treat it, mishandle it a little bit, and it goes to the contracted state. Now, so what we know about the structure? So we actually know the structure of the particle in extended and contracted state, and so we know the structure now of both ends. So not quite about the two states of the base plate, but the sheath and the tube we know for sure. And this is thanks to pretty extensive and heroic work by Peng Ye and Hong Zhou. And so this data has been collected actually on film before direct electron detectors came online, and atomic models have been built. So we know the structure of the pyocin sheath and tube in extended state, and the pyocin sheath in the contracted state. And we are quite certain that the tube doesn't change the structure. So because the symmetry of the tube is the same as the sheath in the extended state. Now, today's talk is all our work that we've done over past maybe 3 years, a bit more even, that describes how this thing goes from this state to this state. So obviously, there's no way I can cover this in 15 minutes. So we'll skip a lot, but I'll try to keep it as understandable as possible. So the question is, can we derive the transformation trajectory from the structure of the end states and free energy considerations? So we approach this contraction process as a physicochemical reaction. We're going to try to use the transition state theory to describe this process. So we have extended metastable high energy state, contracted low energy state, and we have a certain transition state that has a certain activation energy, and its energy higher than the initial state. So if we're able to construct a free energy correctly, then the path with the lowest activation energy is the most probable path, and that's the solution to our problem. So our job is to create a free energy description of the structure, and obviously associate this free energy to the structure in a unique way. This way, we can then relate obviously the structure and the free energy. Now, how to construct the free energy, we need to actually look into the structure a little bit more. So the sheath consists of strands. There's 6 of them. It's a 6-fold symmetric structure and layers. So there's only one subunit, and it makes all of that sheath. It's an oligomeric polymeric structure. And so in the pyocin, the sheath subunit is actually one of the smallest, actually probably the smallest known sheath subunits. It consists of 2 domains: the inner handshake domain and globular protrusion domain. And all other structures, T4, I mean, all other contractile tail-like structures, they actually built on top of this architecture. There's in T4, for example, there are 2 additional domains. There's 1 and 2, and see the pyocin gets away with just only 2 of these domains. In T4, there are 4, as I said, right? So these subunits are connected by these very long arms. So each subunit actually donates an arm to the layer below and to a strand, neighboring strand next to it, and to the layer above. And so we can represent schematically this using this ball and stick representation. So we have this strand connectivity or intra-strand connectivity and inter-strand connectivity. So again, so this is just basically following along these linker arms that connect this sheath subunits. In the contracted state, we have even though the structure is dramatically different, in reality, the connectivity is maintained. So we still have the same strands and we have the same inter-strand connectivity. So in fact, this is actually reminiscent to the sleeves that you put on liquor bottles. So we have this deformative sleeve, so that that can adapt different shapes. But then again, I also have to tell you that the structure, this is universally conserved. All contractile tail-like structures do this. They have this connectivity, and they go from this state to this state, and the symmetry of these structures is very, very similar. Now, so how much the fold of the sheath protein actually changes not much. Right? So this is the expression of extended contracted states. So this goes to the subunit above, and so on. And these pieces actually complement this beta sheet. And here we assemble the subunit from different parts that belong to different subunits. So in fact, this is more or less a rigid unit that represents this ball, that we saw, sphere that we saw in the previous schematic. And from this, so we know the structure of the the subunit in extended and contracted state, but we don't know what's in the middle. So our assumption here, again, it's assumption. It's not a fact. We don't know. So we're going to assume that the structure of the subunit and, as a consequence, the connectivity of the sheath mesh are both preserved throughout the construction event. Because if you think also, if the mesh is deformed, that means one of the strands is pulled out, and obviously the structure probably will not be maintained because the beta sheet is incomplete. So, but this is an assumption. So if we are able to describe this and experiments support what we have here, that means that our assumption is correct. Now, so how we describe the free energy? So we're looking at this complicated connectivity, but again we have this intra and inter-strand connectivity between this inner domain. Outer domain is not shown for clarity. So the total free energy consists of 2 parts, and free energy is an additive function. So there's a free energy associated with the fold of the protein, and there's also free energy associated with the interfaces between these proteins. And this is our total energy, and consists of these 2 parts. But now, if we assume that if the fold is preserved, that means that there's no change of internal energy, or that change can be neglected. So all of the energy is interfacial, and so this we have semi-rigid body approximation. So there's a little bit of deformation in the sheath subunit. We take it into account, but we use this solvent accessible surface area approximation. That's the only way for us to calculate the free energy for a structure of this size. Now, so the actual trajectory will be such that the activation energy is minimal. So that's how free energy works for this spontaneous process. Now, so how big of a structure we need to model? So unfortunately, the structure is pretty big. So yes, asymptotically, we see that's about 12 layers we need to neglect the effects of the two, the top and bottom surface, as you can see. So, all right. So, so what is important here? Yes. So what is important here is that we use the shortest path for the subunit to go from the extended to the contracted state, and it means we can parameterize deformation of every layer using only just one parameter, this contraction fraction. So this is how the sheath subunits move at layer 1, and this is the upper layer. They obviously move differently, but in the system of reference of the subunit itself, it actually moves. It's essentially straight line along the radius and rotates. So that means there is a linear relationship between the angle of turn or turning and radial distance. And this is our contraction. And this is our reaction coordinate. Now, I'm running out of time. So I'm, it's a lot of work. So let me see. So we're to parameterize this contraction reaction, we introduce these harmonic restraints between subunits, and we introduce a drag that allows us to, that compensates for all of the dissipative forces in the system, so that because the upper subunits actually go through a longer distance. And then how we find the activation, minimal activation energy, we actually change the value of the spring constants a little bit, and we try different contraction scenarios. And people who understand structure a little bit, every time you disturb something like this, you introduce a major, obviously, disturbance to the structure. So all of this needs to be rebuilt. So we propagate the perturbation starting from the base plate. We perturb a little bit the bottom layer, and this propagates through the entire structure, and the entire structure needs to be removed or rebuilt. So we have different contraction scenarios. So we have loose springs, we have this delayed contraction, or stiff springs, we have a synchronous contraction. And this allows us to generate different free energy profiles. And then we find an area where the activation is at a minimum. And this is our most probable contraction scenario, but there is more. We need to extrapolate it to the full length structure. Just want to show you this beautiful animations. So again, these are not chimera morphs. These are real trajectories. This is the synchronous trajectory where all the subunits move in sync. So these are very stiff springs, and this is much delayed, or maybe one of the best contraction trajectories. You probably see how the strands play out, and this gives us much lower activation energy. So I'm going to skip a little bit. So we can extrapolate to the full length structure, and this allows us to derive the structure of the transition state. So we know now the contraction fraction of the lower layer of the sheath, and this is 0.84. That means the base plate is almost completely contracted. We can calculate the wavelengths of contraction. So what is the structure of this pyocin particle, where the bottom layer is most contracted and upper layer is still not contracted, and it's actually equal exactly the length of the particle. And what about the experiment? I'm telling you, we did it all. So we can measure the heat that's released during contraction. We can measure the activation energy using Arrhenius approximation. So each of these actually curves. It takes about an hour, and it's repeated so many times. So this is a gazillion hours actually spent on. So each point is like several days of work here. Now as you see in the transition state, there's there's a distance between the strands, and these arms actually really stretched out. We can make and that means that if we make them longer, most likely we lower the activation energy, and that's indeed the case. And if actually particles are functional and so on. But let me tell you. So also we collaborated with Simon Schuring, and they did this state-of-the-art actually pooling of this contracted pyocin sheath, and they measured the spring constant of this guy. So this is, you find the contracted sheath, put the needle AFM needle tip in it, and pull and measure the spring constant. Absolutely remarkable. So this is actually the most remarkable, most important slide, and we can talk actually about an hour just discussing this slide. And so this is we plot the free energy of the system versus tube displacement. So this is a logarithmic scale. As you see, the transition state actually takes place actually fairly early in the this contraction trajectory. Here's the wavelength state where this is most contracted and this is most extended, 2 minutes, actually 1 minute, 30 seconds. And this is finally the contracted state. So if you look at this, so where we can calculate the force, right? So wherever you have this very steep angle on the free energy or any sort of energy curve that you take a gradient, this is where the force is at the maximum. And here is the evolution of curve, because this is a derivative of that. As you see, up to the point where the tube doesn't, or spike doesn't contract the membrane, the force is pretty low. But as soon as the tube comes in contact with the membrane, the force starts to go up. And so we like to measure things in champagne bottle pressures, and so I have to tell, so this is about 7 times the champagne bottle pressure, so it's about 35 atmospheres, and this is 70 atmospheres. But again, so this is a micro world, and this is the pressure that's exerted by the spike on the tube on the membrane. And all phages actually do this. So you see they don't need to develop a force early in the contraction trajectories. They develop much later. So there's always this about 30 angstrom distance between the base plate and the membrane, and that's exactly what our theory shows. That there's no force developed, and then much force developed much later. So no conclusions. So this is published fairly recently, and I really encourage you to go and try to read it now. This is since you now have this primer. So who did the work? Alec has been a driving force in the theory. So Nikolai did all experimental work. And Funk experimental biophysics work, and Funk did this remarkable AFM pooling experiment. All right, guys. Thank you very much. Sorry for going a little bit over time, but there was more than 3 years of work. Thank you.